हेलो एवरीवन, दिस इज मी सर्वेश रिप्रेजेंटिंग पर्सनल ट्यूटर्स। लास्ट वीडियो में हम लोगों ने डिस्कस किया था अबाउट द सेल एंड द सेल थ्योरी। सो we have discussed that cell is the structural and functional unit of life, but how it is structural or how it is functional. See, cell is the only thing, it is the simplest thing which could perform the functional activities which could consider anyone as living. Just example like metabolism. Cells are responsible for the metabolism of any living beings. So द सेल इज लाइक दैट कि अब सेल है, उसके अंदर कुछ कॉन्स्टिट्यूएंट्स होंगे, उन कॉन्स्टिट्यूएंट्स को हम लोग सेल ऑर्गेनल्स बोलते हैं। हर एक ऑर्गेनल का अपना एक स्पेसिफिक फंक्शन होता है बाय विच दे ट्राई टू मेंटेन द इंटीग्रिटी ऑफ द लाइफ। सो द मोस्ट इंपॉर्टेंट सेल ऑर्गेनल्स आर number one is nucleus. Here we will discuss only about the important cell organelles. So nucleus is the main unit of the cell which metabolizes the cell या which controls या which regulates the cell, which regulates the cell functioning. So the nucleus is the only thing which regulates the cell functioning. How? We all know that nucleus has DNA, but is it true for all cells? Yeah, it is true that nucleus have DNA in all cells, but is there nucleus in all cells? No. We have discussed in our previous video that prokaryotes lack differentiated nucleus. Their nucleus is just a simple part of chromosome over there, not chromosome exactly, the strands of DNA coiled in a super coiled manner. So nucleus is the function which regulates the cell. We are here to discuss about the eukaryotic nucleus. Like in prokaryotes, nucleus doesn't have any constitutional units. It only, suppose this is your prokaryotic cell, here it is the nucleus, it is coiled, super coiled, where all the bases are present and everything is present. So in eukaryotic, the nucleus arrangement is not like that. In eukaryotes, the nucleus is covered with a double membrane cell nuclear membrane. Double membrane nucleus covering is present. Here there are certain pores, nuclear pores, which allows the transport of materials inside to outside of the nucleus or outside to inside of the nucleus. So in nucleus there are many different structures. The basic thing is the basic function of nuclear membrane is that it separates, suppose this is a cell, these are the cytoplasm which is filled inside the cell, which constitutes mainly of water and then the certain minerals present, certain minerals, ions. So the nuclear membrane, the double membrane nuclear membrane separates the nucleus from the cytoplasm, and the fluid which is present inside the nucleus will be considered as nucleoplasm. Inside the nucleus or nucleoplasm, in nucleoplasm there is a particular structure known as nucleolus. This nucleolus is the basically ribosomal factory of this cell, which produces the rRNA. We will discuss it about in our further lectures. So basically nucleolus is the unit which is the ribosomal factory and which is responsible. You can directly relate it to protein synthesis, because ribosomes are there to synthesize proteins. The main function of ribosomes are to synthesize the proteins. We will discuss in details when we will continue with the molecular basis of inheritance. So this is nucleolus, which along with nucleolus, some chromatin fibers, which is thread like structure, they are present in nucleus. These thread like fibers, they are actually invisible to our eyes, but it appears like a thread-like structure when we visit it on the microscope. There is a string with beads kind of structure. However, during cell division, this chromatin fibers, they compact, they condenses, and they will form chromosomes over there. In chromosome, this central part is known as the centromere, centrosome, while these all attachments are known as chromatids. So this is nucleus, which contains the chromatin fiber. Now the packaging of DNA is different in eukaryotes as compared to prokaryotes. We had seen in prokaryotes that the nucleus is simply, there is no nucleus, the DNA is simply coiled in a particular fashion, while in eukaryotes it is not like it is coiled with particular fashion. If we look at the structure of DNA, DNAs are negatively charged particles due to presence of OH ions and the phosphate ions. So in DNA, which are negatively charged, they are associated with histone proteins, histone proteins are the positively charged proteins, so their association is comparatively stronger with the proteins and they are properly arranged. Now what is the functioning of arrangement? We'll discuss it on our molecular basis of inheritance chapter. So these are all histone proteins. Actually the DNA is being it is a histone octamer. The main function of histone octamer, or basically histone octamer, is what it is the constitutional units of proteins. Histone octamer is composed of 8 proteins, 2 of different kinds. Like these are 4 kinds of proteins, and each kind of protein has 2 proteins. So basically H1A, 2 proteins of H1A are the most important proteins in the histone octamer, which facilitates the linking. This is H1A. And the DNA is wrapped over it, the whole histone octamer, and along with histone octamer there are some NHC proteins as well. So the NHC stands for non-histone chromosomal protein. So it stands for basically non-histone chromosomal proteins. So these NHC proteins along with histone octamer facilitates the binding of DNA, which is responsible for our functioning. So this is the complete structure of nucleus, where we will have to study the nuclear membrane, the nucleoplasm, and the chromatin threads. Chromatin threads are the pair of DNA, and as you know, DNA is the most important thing for a life, because as it contains the information regarding the life. So that's it for this video. We'll discuss further more cell organelles in our next video. I hope you continue with our channel. Thanks for liking, thanks for subscribing. Please give us your love like this. Like, share and subscribe. Thank you.